Soluble oligomers of amyloidogenic proteins, through their cytotoxic properties, are believed to underlie the pathophysiology of neurodegenerative disease. These oligomers form and dissociate continuously when in solution, a fact that complicates the structural studies that are critical for developing inhibitory molecules. To circumvent this problem, photo-induced cross-linking of unmodified proteins, or PICUP, is used. This method involves the exposure of disaggregated and solubilized amyloidogenic peptides to cross-linking reagents and a 1-second pulse of intense light to covalently link dimers and oligomers. The oligomer size distribution can then be analyzed via standard biochemical techniques.
Hi, I'm Farid Rahimi from the Bitan laboratory in the Department of Neurology at the University of California in Los Angeles.
I am Panchanan Maiti from the Bitan laboratory.
Today we will show you a procedure for photo-induced cross-linking of unmodified proteins.
We use this procedure in our laboratory to generate stabilized amyloid beta protein oligomers and study the effect of structural modifications on the oligomer size distribution.
This procedure was initially published by Fancy and Kolodek to study stable protein complexes. The method was applied later by Bitan and others to quantitative study of metastable amyloid protein assemblies, including a beta, prion, and alpha-synuclein.
So let's get started.
To begin the protocol, hexafluoroisopropanol, or HFIP, which has been pre-chilled on ice, is added.